Good evening and welcome to Dreamy Academy. Tonight, we begin a journey not just through pages or screens, but through the very fabric of existence itself. A quiet exploration of biochemistry, the hidden language of life. As we begin, I'd love to know where you're watching from. Your city, your country, perhaps even what the night sky looks like outside your window. If you enjoy these peaceful explorations of science, please take a moment to like the video and subscribe. It truly helps this little corner of quiet curiosity grow. To those already subscribed, thank you sincerely for being part of this slow and thoughtful adventure. Right now, as you listen, forces older than mountains are shaping the atoms in your bones, in the air you breathe, in the gentle beat of your heart, every breath you take, every thought that forms, every sensation that whispers across your skin is a symphony of atoms and molecules interacting, a cosmic dance happening on scales too small to see, yet too profound to ignore. You are a universe of intricate chemistry, a temporary arrangement of ancient matter that has, through billions of years of cosmic alchemy and biological refinement, learned to contemplate its own existence. It's a beautiful paradox. We are made of the same dust as the stars, yet we possess the capacity to understand the processes that forged us. This journey into biochemistry is not just about understanding the mechanisms of life. It's about recognizing the profound, quiet wonder of being alive, of being a part of this vast interconnected tapestry. We'll drift through the hidden mechanisms that allow life to emerge from mere chemistry, unraveling the elegance of its design, one gentle truth at a time. The brain, it turns out, finds a deep peace in this kind of understanding, a quiet satisfaction in tracing the patterns of the universe as they unfold within us. Consider for a moment the ordinary miracle of a single breath. It is a simple rhythmic act, often unnoticed, yet within that gentle inhalation you draw in a vast invisible crowd of atoms, billions upon billions of them. These aren't just any atoms, they are ancient travellers, particles that have existed since the very early moments of our universe. Some may have once flowed through the lungs of a dinosaur, others perhaps drifted in the atmosphere of a nascent Earth, or even formed part of a distant star before being scattered across the cosmos. This continuous, quiet exchange of matter connects us not only to every living thing that has ever been, but to the deep time of the universe itself. To understand the intricate dance of biochemistry, to truly appreciate the wonder of life, we must first step back, far back, to the most fundamental building blocks of all, the atom. Let's begin by considering what everything around us, and indeed what we are made of. The answer, at its most basic, is matter, and matter, in turn, is composed of tiny fundamental units we call atoms. For a long time, the atom was imagined as a hard, indivisible sphere, a tiny billiard ball. But like many initial intuitions about the universe, this picture, though a useful starting point, is far from the full, elegant truth. Imagine instead a vast empty space, almost entirely void, yet within it a tiny, incredibly dense heart, and around that heart a shimmering, energetic cloud of probability. This is a more accurate and perhaps more wondrous way to envision an atom. At the very centre of this miniature universe, we find the nucleus, a compact, positively charged core. Within the nucleus reside two types of particles: protons, which carry a positive electrical charge, and neutrons, which carry no charge at all. They are bound together by the strongest force known in nature, the strong nuclear force, a force so potent it can only be felt over incredibly short distances, holding these tiny components in a tight embrace. The number of protons in an atom's nucleus is its defining characteristic. It's what gives an atom its identity. This number is called the atomic number, and it dictates what element that atom is. An atom with 1 proton is always hydrogen. An atom with 6 protons is always carbon. An atom with 8 protons is always oxygen. These elements are the fundamental alphabet of the universe, each with its unique chemical personality. Surrounding this dense, central nucleus, like a soft, diffuse whisper, are electrons. These particles carry a negative electrical charge, balancing the positive charge of the protons in a neutral atom. But the electrons don't orbit the nucleus like planets around a sun, as we often visualize. Instead, they exist in what we call orbitals, regions of space where they are most likely to be found. Think of it less as a fixed path and more as a probability cloud, a 3-dimensional wave of potential. An atom, then, is essentially an enormous amount of empty space, with a tiny, dense core and a fuzzy, energetic cloud of possibility where the electrons reside. The nucleus accounts for nearly all of the atom's mass, over 99.9%, but only a minuscule fraction of its volume, perhaps 1 part in 10,000 trillion. If an atom were expanded to the size of a cathedral, its nucleus would be no bigger than a fly resting in the center, and the electrons would be like motes of dust, almost impossibly small, dancing in the vast empty space. It is these electrons, specifically those in the outermost regions of the atom, that orchestrate the magnificent ballet of chemistry. Atoms are not content to exist in isolation. They yearn for stability. This yearning often translates to a desire to have a full outer shell of electrons, a state of energetic contentment. To achieve this, atoms will interact with each other in various ways, forming what we call chemical bonds. These bonds are not physical ropes or hooks. They are powerful electromagnetic attractions, invisible forces that hold atoms together to create molecules. Imagine 2 hydrogen atoms, each with 1 proton and 1 electron. Their outermost shell, the first shell, to hold 2 electrons to be stable. Neither hydrogen atom has enough electrons on its own, so they come together and share their electrons, forming a covalent bond. Each hydrogen atom now effectively "3" 2 electrons in its outer shell, and both are content. The result is a hydrogen molecule, H2. This sharing of electrons is one of the most fundamental ways atoms connect, and it creates incredibly strong, stable bonds. Water, that essential solvent of life, is formed by 2 hydrogen atoms covalently bonded to 1 oxygen atom. Oxygen, with its 8 protons, has 6 electrons in its outer shell and 8. By sharing with 2 hydrogens, it achieves that stability, and in doing so, creates one of the most remarkable molecules in the universe. The specific angles and lengths of these covalent bonds give molecules their unique shapes, and as we will see, shape is everything in biochemistry. Another way atoms achieve stability is by completely transferring electrons, forming what are known as ionic bonds. Picture a sodium atom with its 11 protons. It has 1 electron in its outermost shell, and it would be much more stable if it could simply get rid of that single electron, leaving a full inner shell. Now imagine a chlorine atom with 17 protons. It has 7 electrons in its outermost shell and 1 more to complete its shell. It's a perfect match. The sodium atom readily gives up its electron to the chlorine atom. When sodium loses an electron, it becomes positively charged, an ion. When chlorine gains an electron, it becomes negatively charged, also an ion. And what happens when a positive charge meets a negative charge? They attract each other strongly. This electrostatic attraction is an ionic bond, and it's how table salt, sodium chloride, is formed. These bonds are strong, but unlike covalent bonds, they don't have a fixed direction. The attraction is omnidirectional. These two types of bonds, covalent and ionic, along with other gentler interactions we'll explore later, are the universe's fundamental tools for constructing complexity. From a mere handful of stable elements, primarily hydrogen, carbon, oxygen, nitrogen, phosphorus, and sulfur, the 6 most abundant elements in living organisms, life has learned to build an astonishing array of molecules. Carbon, in particular, is a master architect. With its 4 electrons in its outer shell, it can form 4 strong covalent bonds with other atoms, including other carbon atoms. This unique ability allows carbon to form long, stable chains, rings, and intricate branching structures, creating the vast molecular skeletons upon which life is built. The universe, in its elegant simplicity, gave us an alphabet of elements, each with its own quiet personality, defined by the number of protons in its heart. From this alphabet, and from the fundamental drive of electrons to find stability, molecules are formed. These molecules are not static objects. They are dynamic, vibrating structures, constantly interacting, breaking, and reforming. They are the words, sentences, and paragraphs of the universe's oldest story. Every protein, every strand of DNA, every sugar molecule in your body is a testament to the elegant rules of atomic interaction, a complex sculpture made of electromagnetic forces. There's a particular comfort in understanding that the very essence of our being, the very mechanisms that allow us to think and feel, are rooted in these unchanging ancient principles of atomic architecture. The beauty is in the precision, the sheer inevitability of these interactions leading to such profound complexity. So, we've seen how the universe's most basic ingredients, atoms and their electrons, interact through the fundamental forces of attraction and repulsion to form molecules. We've glimpsed the quiet, invisible architecture that underpins all matter, from the simplest hydrogen atom to the intricate structure of a water molecule. But these are just the first whispers of the story. How do these simple building blocks, these letters and words of the chemical alphabet, arrange themselves into the vast, complex, and purposeful structures that define living things? How do they begin to form the long, intricate chains and folded shapes that can store information, catalyze reactions, and build the very machinery of life? That question leads us deeper into the molecular realm, where these elegant rules of bonding begin to sculpt the macromolecules that are the true giants of biochemistry. We left off, didn't we, with a question lingering in the quiet air between us. A profound query, really. How do these humble atomic interactions, the sharing and transferring of electrons, the yearning for stability, evolve into the vast, complex macromolecules that store our very blueprints, catalyze life's ceaseless reactions, and form the intricate machinery of existence? It's a leap, isn't it? From a single carbon atom's 4 graceful bonds to the sprawling elegance of a protein, or the spiraling majesty of DNA. Today, dear friends, we take a step into that grand assembly, into the molecular realm where simple building blocks, like ancient patient masons, begin to construct the towering cathedrals of life. We'll explore how these fundamental elements, our alphabet of existence, begin to spell out words, then sentences, and finally, entire epic poems. This is where the quiet architecture we spoke of in Chapter 1 truly begins to sing, where the individual notes of atoms combine into the rich, resonant chords of biochemistry. Imagine, if you will, a vast and ancient workshop. Here, the master architects of life don't simply create one-off sculptures. No, their genius lies in repetition, in the elegant simplicity of building enormous, intricate structures from countless identical, or nearly identical, small units. This is the secret, the fundamental principle that underpins the construction of nearly all of life's largest molecules. The concept of monomers linking to form polymers. A monomer, in its essence, is a single, small molecular unit. Think of it as a single bead. A polymer, then, is the long, magnificent necklace formed when hundreds, thousands, even millions of these beads are strung together. It's a universal language of construction, a testament to efficiency and elegance. From the simple repetition of a single type of bead, or a subtle variation of a few types, emerges astonishing complexity and diversity. It is a whisper of the cosmos, echoing the fractal beauty found in snowflakes and galaxies alike. And what are these grand structures, these polymers, that form the very fabric of you and me, of every leaf, every fleeting breath of air exhaled by a tree? We classify them into 4 magnificent families, the 4 pillars upon which all life is built: carbohydrates, lipids, proteins, and nucleic acids. Each family has its own unique story, its own particular role in the vibrant, ongoing drama of life, but all share that common thread of assembly from smaller units. Let's unfold them, one by one, with the reverence they deserve. First, we encounter these young carbohydrates. Oh, the sweet architects of energy and structure. Their very name hints at their composition. Quote 6 for carbon and quote 7 for water, as they are essentially carbon atoms adorned with hydrogen and hydroxyl groups. Their simplest form, the monomer, is a monosaccharide, a single sugar unit. Glucose, that ubiquitous molecule, is perhaps the most famous monosaccharide, a humble 6-carbon ring that is the immediate fuel source for nearly every cell on Earth. Fructose, the sweetness of fruit, and galactose, a component of milk sugar, are other familiar faces in this family. But these simple sugars are just the beginning. Like individual notes in a melody, they can link together, through quiet covalent bonds, to form disaccharides, like sucrose, our common table sugar, a union of glucose and fructose, and, more importantly, polysaccharides. The grand polymers of carbohydrates. Consider starch, the energy storage molecule in plants. It's a long, branching chain of glucose units, a vast molecular pantry where plants tuck away their solar earnings for a rainy day, or for our own consumption. When we eat a potato or a grain of rice, we are essentially tapping into these carefully woven glucose reserves. Our bodies, with their own ingenious enzymes, then break these long chains back down into individual glucose molecules, releasing that stored energy to power our thoughts, our movements, our very heartbeat. Then there's glycogen, the animal equivalent of starch, our own internal energy bank, primarily stored in the liver and muscles. It's an even more branched, more intricately woven polysaccharide, designed for rapid access when our bodies demand a quick surge of fuel. Imagine it, a molecular savings account always ready for withdrawal. And for structure, think of cellulose, the very essence of plant cell walls. It's also a polymer of glucose, but with a slight difference in how the glucose units are linked, creating a rigid, linear structure. This subtle twist in bonding is what makes wood strong, what gives vegetables their crunch, and what, for the most part, makes cellulose indigestible for us, passing through as dietary fibre. It is the skeletal framework of the plant world, a quiet, unyielding architecture that allows trees to reach for the sky and leaves to unfurl in the sun. So carbohydrates are not just about sweetness, they are the immediate currency of life's energy and the silent scaffolding that gives form and strength to much of the living world. They are the initial spark, the sturdy beam, the sweet promise of sustenance. Next, we venture into the shimmering, sometimes elusive world of lipids. These molecules are often misunderstood, perhaps unfairly associated with weight gain or dietary concerns. But in the grand tapestry of life, lipids are indispensable, performing roles no other molecule can. Unlike carbohydrates, proteins, and nucleic acids, lipids are not typically polymers of repeating monomer units in the same linear fashion. Instead, their defining characteristic is their insolubility in water. They are hydrophobic. Quote, "This trait, as we shall see, is not a flaw but a profound strength, essential for life's boundaries. The most familiar lipids are perhaps the fats and oils, known chemically as triglycerides. These are formed from two simpler components, a glycerol molecule, a small 3-carbon alcohol, and 3 long chains of fatty acids. These fatty acid chains are long hydrocarbon tails, sometimes straight and saturated, like butter, sometimes bent and unsaturated, like olive oil, their subtle differences dictating their physical properties. Fats are life's long-term energy reservoirs, packing more than twice the energy per gram compared to carbohydrates. They are the deep, silent wells of stored power, insulating us, cushioning our organs, and providing a slow, steady release of fuel. Picture them as the ancient hidden treasuries of energy, waiting patiently to be called upon. But the true marvel of lipids, the architectural masterpiece, lies in the phospholipids. These are similar to triglycerides, but one of the fatty acid chains is replaced by a phosphate group which is hydrophilic. Quote 9. This creates a molecule with a split personality, a hydrophilic head that loves water, and 2 hydrophobic tails that shun it. When placed in water, these phospholipids spontaneously arrange themselves into a bilayer, a double layer where the hydrophobic tails hide inward, shielded from the water, and the hydrophilic heads face outward, embracing the aqueous environment. This phospholipid bilayer, my friends, is the very essence of the cell membrane. It is the whispering boundary, the delicate yet formidable skin that encloses every single cell, defining its inside from its outside. It is a selective gatekeeper, allowing some substances to pass, blocking others, maintaining the precise internal environment necessary for life. Without this lipid architecture, cells would simply dissipate into their surroundings, unable to maintain their intricate internal order. It is the quiet guardian, the silent architect of cellular individuality, And let us not forget the steroids, another class of lipids characterized by their unique 4-ring carbon structure. Cholesterol, often viewed negatively, is in fact a vital component of animal cell membranes, providing fluidity and stability. It is also the precursor for many hormones, those molecular messengers that orchestrate growth, metabolism, and reproduction. From the silent boundaries of cells to the subtle whispers of hormonal communication, lipids are the unsung heroes, the liquid gold of life's intricate machinery. Now we approach the most diverse and arguably the most dynamic of life's macromolecules, the proteins. If carbohydrates are the fuel and the framework, and lipids are the boundaries and reservoirs, then proteins are the master performers, the versatile workhorses, the very engines and architects of every cellular function. They are the molecular dancers, the intricate machines, the master craftsmen that build, break down, transport, defend, and communicate. The monomers of proteins are passenger amino acids. There are 20 different common types of amino acids, each with a central carbon atom bonded to an amino group, a carboxyl group, a hydrogen atom, and a unique side chain, or, quote, 10. It is this R group that gives each amino acid its distinct personality. Some are hydrophilic, some hydrophobic, some acidic, some basic. Imagine these 20 amino acids as a sophisticated alphabet. Just as 26 letters in our English alphabet can form an infinite number of words, sentences, and stories, these 20 amino acids can link together in countless sequences to form long chains called polypeptides. These polypeptides, however, are not yet functional proteins. Their true magic begins when they fold. This folding is a breathtaking ballet, dictated by the sequence of amino acids and their interactions. The polypeptide chain spontaneously twists into local structures, alpha helices and beta sheets, the Hons secondary structure, then contorts further into a unique, intricate 3-dimensional shape, the tertiary structure. Some proteins, like hemoglobin in our blood, even combine multiple folded polypeptide chains into a larger multi-subunit complex, the quaternary structure. It is this precise, exquisite 3-dimensional shape that determines a protein's function. Denature a protein, disrupt its shape, and it loses its ability to perform its task like a key no longer fitting its lock. What do proteins do? The list is astonishingly vast. Enzymes. Perhaps their most famous role. These are biological catalysts speeding up biochemical reactions by millions or billions of times without being consumed themselves. Without enzymes, life's reactions would proceed too slowly to sustain us. They are the tireless facilitators, the invisible hands guiding every metabolic process. Structural proteins. Collagen in our skin, hair, nails, and bones. Keratin in our hair and nails. They provide support, strength, and elasticity, the very scaffolding of our bodies. Transport proteins. Hemoglobin carries oxygen in our blood. Channel proteins in cell membranes ferry substances in and out. They are the diligent couriers, the molecular ferries. Signaling proteins. Hormones like insulin, a protein, and receptor proteins on cell surfaces allow cells to communicate with each other, orchestrating complex responses. They are the messengers, the attentive listeners. Defense proteins. Antibodies, components of our immune system, recognize and neutralize invaders. They are the vigilant sentinels, the protectors. Motor proteins. Actin and myosin, responsible for muscle contraction. They are the movers, the tireless engines of motion. Proteins are the living paradox. Built from a relatively small alphabet, they create an almost infinite library of forms and functions, each perfectly suited for its specific role. They are the very embodiment of the quote 11 we began to explore, transforming simple chemical bonds into the dynamic, purposeful machinery of life itself. Finally, we arrive at the profound and ancient realm of the Sanka, nucleic acids. These are the sacred scrolls, the ancient libraries, the very blueprints and operating manuals of life. They are the molecules that store and transmit the genetic information that makes you, and every living thing precisely what it is. The monomers of nucleic acids are called nucleotides. Each nucleotide is a tripartite structure, a small, elegant assembly consisting of 3 components: a 5-carbon sugar, either deoxyribose in DNA or ribose in RNA, a phosphate group, and a nitrogen-containing base. There are 5 main nitrogenous bases: adenine, A, guanine, G, cytosine, C, thymine, T, found only in DNA, and uracil, U, found only in RNA. These nucleotides link together, forming long chains called pairs polynucleotides. And it is in the structure of these polynucleotides that the magic truly unfolds. DNA, deoxyribonucleic acid. The undisputed star of genetic information typically exists as a double helix. Imagine 2 polynucleotide strands twining around each other like a majestic spiral staircase. The sugar-phosphate backbones form the outer railings, while the nitrogenous bases face inward, forming the quote 12. Here's the crucial part. The bases pair up in a specific way. Adenine, A, always pairs with thymine T, and guanine, G, always pairs with cytosine, C. This base pairing is the fundamental language of genetic information. The sequence of these base pairs along the DNA molecule encodes all the instructions for building and operating an organism. It is the master plan, the complete architectural drawing for life. RNA, ribonucleic acid, while similar, typically exists as a single strand and uses uracil, U, instead of thymine, T. RNA plays various vital roles, acting as a messenger, mRNA, to carry genetic instructions from DNA to the protein-making machinery, as a translator, RNA, to convert that message into amino acid sequences, and as a structural and catalytic component, rRNA, of the ribosomes, the cellular factories where proteins are assembled. RNA is the dynamic interpreter, the diligent worker, the essential bridge between the blueprint and the built structure. Nucleic acids are the ultimate information architects. They are the quiet keepers of history, the silent narrators of evolution, holding within their intricate structures the accumulated wisdom of billions of years of life on Earth. They are the threads that connect us not just to our parents, but to the very first single-celled organisms that grace this planet. So, we have journeyed through the 4 great families of macromolecules: carbohydrates, lipids, proteins, and nucleic acids, each in its own wondrous way, built from simpler units, each playing an indispensable role in the symphony of life. From the immediate energy of a sugar, to the protective embrace of a lipid membrane, to the tireless work of a protein, and the ancient wisdom held within nucleic acids, these are the grand assemblies, the magnificent structures that emerge from the quiet dance of atoms. They are not isolated entities, these macromolecules, but rather a vast interconnected network constantly interacting, communicating and transforming within the bustling metropolis of the cell. They are the very fabric of our being, the tangible manifestation of the quiet wonder of being alive. As we pause here, reflecting on these molecular marvels, let us carry forward this sense of awe, for the journey is far from over. We have glimpsed the blueprints and the building blocks, the architects and their tools. But how do these structures then perform their astonishing feats? How do they collaborate, coordinate, and create the dynamic, self-sustaining miracle that is life? That, my friends, is a story for our next quiet exploration. We've journeyed through the quiet architecture of life, tracing the elemental threads that weave together to form the grand tapestries of macromolecules. We've seen the elegant precision of carbohydrates, the hydrophobic grace of lipids, the intricate folding of proteins, and the profound wisdom encoded within nucleic acids. These are not static monuments, however, but vibrant living entities ceaselessly interacting within the bustling molecular realm of the cell. Now, the question gently arises, how do these magnificent structures move? How do they sing their molecular songs, transforming one into another, building, breaking, and sustaining the delicate flame of existence? Today, we delve into the very heart of this dynamic interplay, into the ceaseless ballet of chemical reactions that defines life itself. Imagine, if you will, a vast unseen theatre within every cell, where countless microscopic dramas unfold each moment. Atoms, those ancient travellers we first encountered, are constantly rearranging their alliances, breaking old bonds and forging new ones. This fundamental act of rearrangement is a chemical reaction, a whisper of transformation, a shift in the molecular landscape. Consider the simplest breath you take. Oxygen molecules, those eager electron seekers, meet with fuel molecules, and in a flurry of bond-breaking and bond-making, energy is released, and new molecules, like carbon dioxide and water, are born. It is a constant, exquisite conversation, a dialogue between matter and energy. Each of these molecular conversations carries a subtle energetic signature. Some reactions, like the gentle cascade of water flowing downhill, release energy into their surroundings. These are these exergonic reactions. The energy-releasing moments, often associated with the breakdown of larger, more complex molecules into simpler ones, a process we call catabolism. Think of a grand, ancient tree slowly returning its nutrients to the soil, or the careful dismantling of a complex machine into its constituent parts. There is a sense of release, of potential being actualized. Conversely, other reactions are like pushing a stone uphill, requiring a continuous input of energy to proceed. These are the endergonic reactions. The energy-requiring moments typically involved in building larger, more intricate molecules from simpler precursors, a process known as anabolism. Imagine the painstaking construction of a majestic cathedral brick by brick, beam by beam, each step demanding effort and energy. Life, in its breathtaking complexity, is a constant dance between these two forces, the gentle dismantling that releases energy, and the careful construction that harnesses it. Together, this intricate network of breaking down and building up is known as metabolism, the grand symphony of chemical transformations that animates every living cell. But here lies a fascinating paradox. Many of these energy-releasing reactions, though thermodynamically favourable, do not simply happen spontaneously at the gentle temperatures and pressures found within a living cell. There is often an invisible barrier, a molecular hill that must be climbed before the downhill slide can begin. We call this the activation energy, the initial spark, the gentle nudge, the minimum energy required to initiate a chemical reaction. Imagine pushing a boulder to the edge of a cliff. It won't roll down on its own until you've expended enough effort to get it over the initial hump. Without this initial push, the boulder remains stubbornly still. In the same way, molecules often need a specific amount of energy to reach a transient, unstable state, a transition state, before they can rearrange into their new forms. Without this, life's essential reactions would proceed at a glacial pace, far too slow to sustain the vibrant dynamism we witness. This is where the true unsung heroes of the cellular stage make their grand entrance, the enzymes. If the activation energy is a daunting mountain pass, then enzymes are the master road builders, carving gentle slopes and tunnels through the formidable terrain, making the journey swift and effortless. Enzymes are, almost without exception, proteins, those marvels of intricate folding we explored in our last chapter. They are biological catalysts, meaning they dramatically accelerate the rate of chemical reactions without being consumed in the process themselves. They are the tireless choreographers of the molecular ballet, guiding their partners through their precise movements, ensuring every step is taken with grace and speed. What makes enzymes so extraordinary? It is their exquisite specificity. Each enzyme, like a perfectly crafted key, is designed to interact with a particular molecule or a small group of molecules known as its substrate. This interaction often follows a Jan quote 17 model, where the substrate fits precisely into a specific region on the enzyme called the active site. Or perhaps even more beautifully, it's an induced fit model, where the enzyme gently adjusts its shape upon binding to the substrate, embracing it in a warm, facilitating clasp. Within this intimate embrace, the enzyme works its magic. It might strain specific bonds within the substrate, bringing reactive groups into perfect alignment, or creating a localized environment that favors the reaction, by subtly manipulating the molecular landscape. The enzyme effectively lowers the activation energy, allowing the reaction to proceed millions of times faster than it would otherwise. Without these magnificent molecular artisans, the metabolic symphony would fall silent, and the very concept of life as we know it would cease to exist. But even with enzymes clearing the path, where does the energy come from to power the uphill anabolic reactions? How do cells pay for the constant building and maintenance that defines their existence? Here, we encounter the universal energy currency of life, ATPUE, or adenosine triphosphate. Imagine ATP as the cell's tiny rechargeable battery, a compact packet of readily available energy. Structurally, ATP is a marvel of elegant design. It consists of an adenine base, a ribose sugar, and most importantly, 3 phosphate groups linked together in a chain. It is in the bonds between these phosphate groups, particularly the terminal one, that a significant amount of chemical potential energy is stored. When a cell needs energy, it performs a simple yet profound act. It hydrolyzes, or breaks, that terminal phosphate bond, releasing a phosphate group and transforming ATP into ADP, adenosine diphosphate. This breaking of the bond is like releasing a compressed spring, liberating a burst of energy that can be immediately harnessed to power endergonic reactions. This process is called energy coupling. The energy released from ATP hydrolysis is used to drive a reaction that would otherwise require an input of energy. It's a beautifully efficient system, ensuring that energy is captured from exergonic catabolic reactions and then precisely delivered to fuel endergonic anabolic reactions, preventing wasteful dissipation. Think of it as a constant cycle of charging and discharging, a ceaseless flow of energy that sustains every cellular activity, from muscle contraction to the synthesis of a new protein. As we widen our gaze from individual reactions, we begin to perceive an even grander design: metabolic pathways. No single reaction truly stands alone in the cell. Instead, they are woven into intricate interconnected sequences, like the tributaries of a vast river system. A metabolic pathway is a series of chemical reactions, each catalyzed by a specific enzyme, where the product of one reaction becomes the substrate for the next. Imagine a molecular assembly line, where each station performs a precise transformation, meticulously guided by its own enzyme, ultimately leading to a final product. There are two main types of these pathways, reflecting the catabolic and anabolic forces we discussed earlier. Catabolic pathways are the routes of breakdown, where complex molecules are systematically dismantled to release energy and simpler building blocks. The most renowned example is cellular respiration, the pathway by which glucose, our cellular fuel, is broken down step by step, extracting its stored energy to generate ATP, like a power plant converting fuel into electricity. It is a marvel of controlled combustion, releasing energy in small, manageable packets rather than a single explosive burst. Conversely, anabolic pathways are the routes of synthesis, where the cell expends energy, often in the form of ATP, to construct complex molecules from simpler precursors. Think of photosynthesis, where plants harness the sun's energy to build glucose from carbon dioxide and water, a breathtaking act of molecular architecture. Or consider the intricate pathways that assemble amino acids into proteins or nucleotides into DNA. These are the creative forces of life, transforming simple matter into the breathtaking complexity of living forms. What is truly astonishing is the exquisite regulation of these pathways. Cells are not simply chaotic factories, they are finely tuned, self-regulating systems. Enzymes at key junctures act as molecular switches, turning pathways on or off, speeding them up or slowing them down in response to the cell's immediate needs. This intricate control ensures that resources are not wasted, that energy is efficiently managed, and that the delicate balance, or homeostasis, of the internal environment is maintained. It is a testament to billions of years of evolutionary refinement, a silent wisdom embedded within the very fabric of life. So, as we bring this chapter to a gentle close, let us pause and reflect on the profound beauty of this dynamic cellular world. We have moved beyond the static blueprint of macromolecules to witness the constant, vibrant flux of their interactions. We've seen the elegant necessity of activation energy, the tireless dedication of enzymes, the universal pulse of ATP, and the orchestral harmony of metabolic pathways. These are not just abstract concepts, they are the very engines of life, collaborating in a ceaseless, awe-inspiring dance that underpins every breath, every thought, every beat of your heart. The cell, then, is not merely a collection of molecules, but a living, breathing testament to the power of transformation, a miniature universe of ceaseless activity, all orchestrated with an elegance that humbles the most sophisticated human engineering. In this constant flow of energy and matter, we find the quiet wonder of being alive, a profound interconnectedness that stretches from the single chemical bond to the vast, self-sustaining miracle of life itself. What magnificent secrets will these dynamic systems reveal next? We shall see as we continue our journey into the wondrous realm of biochemistry. We have journeyed through the intricate dances of atoms, witnessed the grand assembly of macromolecules, and marveled at the dynamic ebb and flow of metabolic pathways, where energy is sculpted and transformed. We've seen how reactions ignite and subside, how enzymes orchestrate the ballet of chemical change, and how ATP, the shimmering currency of life, powers the very engine of existence. But where, you might ask, does this magnificent symphony truly play out? In what hallowed hall do these countless precise interactions occur, orchestrated with such breathtaking precision? Let us now turn our gaze to the most fundamental and wondrous stage of all, the cell. Imagine, if you will, not merely a microscopic bag of chemicals, but a miniature universe, self-contained and self-sustaining, a bustling city of purpose and profound design. It is here, within the quiet confines of this single, living entity, that the biochemistry we have explored thus far takes on its most vibrant, tangible form. The cell is the canvas upon which life's masterpiece is painted, the crucible where the raw elements of matter are transmuted into the living essence of being. Our exploration begins at the very boundary of this microcosmos, its delicate yet formidable outer skin, the cell membrane. Consider it the wise guardian, the discerning gatekeeper, the sentient envelope that cradles the cell's precious interior while maintaining an intimate yet selective dialogue with the world beyond. This membrane, as we briefly touched upon with lipids, is a shimmering bilayer of phospholipids, their hydrophilic heads facing the watery environments both inside and outside the cell, and their hydrophobic tails tucked safely within, away from the aqueous embrace. It is a structure both fluid and stable, a dynamic mosaic studded with proteins that act as channels, pumps, receptors, and identifiers. What an extraordinary feat of molecular architecture this membrane represents. It is not merely a wall, but a living, breathing interface. It determines what enters and what exits, a constant negotiation of nutrients flowing in and wastes flowing out, maintaining the delicate internal equilibrium we call homeostasis. Through its embedded proteins, it receives signals from other cells, interpreting messages that dictate growth, division, and function, allowing cells to communicate in a silent, intricate language. This shimmering barrier, then, is not just a boundary, it is a declaration of individuality, a statement of existence, and a testament to the elegant simplicity that underpins profound complexity. Now, step with me across this threshold, and let us immerse ourselves in the cell's inner landscape. Once inside, we find ourselves in the cytosol, a jelly-like, aqueous solution that fills the cellular space. Think of it as the cell's bustling inner sea, a vibrant fluid matrix where countless metabolic pathways, those intricate series of reactions we discussed, unfold. Here, many of the initial steps of glucose breakdown, the very first whispers of energy extraction, take place. It is a space of constant motion, of molecules diffusing, colliding, and reacting, a dynamic liquid realm where the foundational work of life begins. Yet as we gaze deeper into this cellular ocean, we perceive structures, like islands or continents, each with its own distinct form and purpose. These are the organelles, the specialized compartments that bestow upon their eukaryotic cell its remarkable efficiency and organizational prowess. Imagine a vast factory, not as one sprawling, undifferentiated space, but as a meticulously organized complex with distinct departments for manufacturing, energy production, waste management, and communication. This is the eukaryotic cell, a universe of specialized labor, where each organelle contributes its unique wisdom to the collective life of the whole. At the very heart of this cellular city, often occupying a prominent central position, resides the nucleus. This is the cell's command center, its sacred library, housing the entirety of the cell's genetic blueprint, the DNA, encased within its own double membrane, the nuclear envelope, which is punctuated by pores allowing selective communication with the cytosol. The nucleus is the sanctum, where the precious scrolls of heredity are carefully stored and meticulously guarded. Here, the process of transcription occurs, where segments of DNA are copied into messenger RNA, mRNA, carrying the instructions for building proteins out into the wider cellular realm. It is a place of profound quietude and immense power, where the very essence of what the cell is and what it can become is encoded. Moving outwards from this central archive, we encounter a vast interconnected network of membranes that permeates much of the cytoplasm, the unendoplasmic reticulum, ER. Picture it as a labyrinthine system of interconnected sacs and tubules, a sophisticated internal transportation and manufacturing hub. We distinguish between two forms, each with its own specialized tasks. First, there is the rough endoplasmic reticulum, RER, so named because its outer surface is studded with tiny granular particles, ribosomes. These ribosomes, they are the cell's tireless protein builders, the molecular artisans who translate the genetic code carried by mRNA into the specific sequences of amino acids that form proteins, as we explored in Chapter 2. On the RER, these ribosomes synthesize proteins destined for secretion from the cell, or for insertion into membranes, or for delivery to other organelles. Inside the RER's lumen, these nascent proteins begin their intricate journey of folding, guided by chaperones, undergoing modifications, and embarking on a rigorous quality control check. It is a bustling protein factory, ensuring that each protein emerges in its correct, three-dimensional form, ready for its specific role. Adjacent to the RR, and often continuous with it, lies the smooth endoplasmic reticulum, SER. This region lacks ribosomes, giving it a smoother appearance, but its functions are no less vital. The SER is a versatile workshop, engaged in the synthesis of lipids, including phospholipids and steroids, those essential components we encountered in Chapter 2, forming membranes and hormones. It also plays a crucial role in detoxification, metabolizing various drugs and harmful substances, transforming them into more water-soluble forms for easier excretion. Furthermore, in muscle cells, the SER, often called the sarcoplasmic reticulum, is a vital reservoir for calcium ions, essential for muscle contraction. The ER, in its dual forms, truly acts as the cell's internal scaffolding, its manufacturing plant, and its chemical processing unit. As proteins and lipids are synthesized in the ER, they often embark on a further journey of refinement and sorting. This leads us to the Golgi apparatus, sometimes referred to as the Golgi complex or Golgi body. Imagine it as the cell's sophisticated post office or shipping centre. It consists of a stack of flattened membranous sacs called cisternae, which receive vesicles, small membrane-bound sacs, budding off from the ER. Within the Golgi, proteins and lipids are further modified, sorted and packaged into new vesicles, destined for various cellular locations or for secretion outside the cell. It's a marvel of molecular logistics, ensuring that each molecular package reaches its correct address, a testament to the cell's impeccable organizational skills. And what of the cell's energy demands? Life, as we know, is an energy-intensive endeavor, constantly requiring ATP to fuel its myriad processes. This brings us to the mighty mitochondria, often described as the powerhouses of the cell. These fascinating organelles, each encased in a double membrane with the inner membrane folded into cristae, are the primary sites of cellular respiration. Here, the stored chemical energy in glucose and other fuel molecules is efficiently converted into ATP, the universal energy currency we encountered in Chapter 3. With their own small circular DNA and ribosomes, mitochondria bear the intriguing hallmarks of ancient bacteria, suggesting a profound symbiotic origin. They are the tiny furnaces of life, tirelessly generating the energy that allows the cell to grow, move, synthesize, and simply be. But even the most efficient city generates waste, and cells are no exception. For this we have the lysosomes, the cell's recycling centers or digestive sacs. These spherical organelles contain powerful hydrolytic enzymes capable of breaking down worn-out cellular components, invading bacteria and other cellular debris into their simpler building blocks. Once broken down, these components can often be recycled, a beautiful example of cellular economy and sustainability. Similarly, peroxisomes are small, membrane-bound organelles involved in various metabolic processes, including the breakdown of fatty acids and the detoxification of harmful substances, often generating hydrogen peroxide as a byproduct, which they then convert into harmless water and oxygen. Beneath all this bustling activity, providing structure, support, and pathways for movement, lies the dynamic cytoskeleton. Consider it the cell's internal scaffolding, its intricate network of protein filaments, microtubules, microfilaments, and intermediate filaments. This isn't a static framework. It's a constantly remodeling dynamic system that gives the cell its shape, allows for movement, like cell crawling or muscle contraction, and provides tracks along which organelles and vesicles are transported. It is the cell's internal highway system, its architectural backbone, and its engine for locomotion, a testament to the cell's incredible adaptability and internal dynamism. As we gently withdraw our gaze from this inner cosmos, do you not feel a profound sense of awe? Each organelle, a specialized compartment with its unique biochemical machinery, does not operate in isolation. Instead, they are profoundly interconnected, a cooperative society working in seamless harmony. Proteins synthesized on the RER are trafficked to the Golgi, packaged into vesicles, and transported along the cytoskeleton. Mitochondria provide the ATP that powers these movements and syntheses. The nucleus dictates the entire symphony. It is a dance of exquisite coordination, a testament to millions of years of evolutionary refinement. The cell, then, is not merely a collection of parts, but a living, breathing entity, a microcosm of the grand tapestry of life itself. It is the fundamental unit where all the principles of biochemistry we have explored, the bonds, the macromolecules, the energy transformations, the enzymatic catalysis, converge into the miracle of a living system. This tiny, complex world, often invisible to the naked eye, holds within its boundaries the very essence of existence, a quiet wonder humming with purpose and profound biochemical elegance. We are just beginning to appreciate the depth of its wisdom, a wisdom echoed in every breath we take, every thought we think, every beat of our hearts. We have journeyed together, haven't we, through the intricate landscapes of existence, from the ancient dance of atoms to the breathtaking organization within a single cell. We've marveled at the cell membrane, that whisper-thin yet profoundly powerful guardian, and peered into the bustling world of organelles, each a specialist in a grand symphony of life. We've come to see the cell not merely as a boundary, but as a miniature universe, perfectly orchestrated, a testament to quiet wonder and profound biochemical elegance. But as we pause at this threshold, a new compelling question arises, one that beckons us deeper into the cell's luminous heart. How does this magnificent entity, this teeming metropolis of molecules and membranes, truly know what to do? How does it receive the myriad messages from its environment, both near and far interpret their subtle nuances, and then respond with such precision and grace? How do the billions of cells within a complex organism, like our own bodies, coordinate their actions, developing, growing, healing, and thriving as one harmonious whole? Consider, if you will, the cell not just as a dwelling, but as a sentient listener and a thoughtful speaker in the vast, ongoing conversation of life. It is an entity constantly attuned, constantly interpreting the silent language of its surroundings. Imagine a quiet garden where each bloom, each blade of grass, each unseen root system is not isolated but intimately connected, sharing whispers carried on the breeze, signals transmitted through the soil. So too are our cells, engaged in a ceaseless dialogue, a ballet of information exchange that ensures life's continuity and its astonishing adaptability. This intricate art of receiving, interpreting, and responding to signals is what we call cellular signaling or cell communication. It is, in essence, the nervous system of the microscopic world, allowing a cell to perceive changes, to adapt, and to collaborate. Without this profound ability, life as we know it, from the simplest bacterium navigating a nutrient gradient to the most complex thoughts forming within our minds, would simply cease to be. Let us begin by understanding the fundamental players in this exquisite drama. Every message, every signal begins with a ligand. Think of a ligand as a molecular whisper, a specific chemical key. These keys can take many forms. They might be tiny ions, small organic molecules, gases, or even complex proteins. What makes them special is their ability to bind with remarkable specificity to another molecule within or on the surface of the target cell. This second molecule is the receptor, the lock designed precisely for that particular key. These receptors are most often proteins carefully sculpted by evolution to recognize and embrace their unique ligand. They are the cell's antennae, its ears, its eyes, constantly scanning the environment for the specific signals meant for them. This binding, this moment of recognition between ligand and receptor, is the very first step in cellular communication, a moment of profound significance, that sets in motion a cascade of events within the cell. Now, the distance over which these cellular conversations unfold can vary dramatically, much like the different ways we might communicate with one another. Sometimes, a cell might speak directly to itself, a quiet internal monologue of self-regulation. This is autocrine signaling, where a cell releases a signaling molecule that then binds to receptors on its own surface, influencing its own behavior. It's a way for a cell to fine-tune its own activities, perhaps to ensure proper growth or to regulate a specific internal process. Then there's the more common form of local gossip, if you will, known as paracrine signaling. Here, cells release signaling molecules that act on nearby neighboring cells. Think of a small community where news travels quickly from house to house. Neurotransmitters, those swift messengers of our nervous system, are a beautiful example of paracrine signaling, bridging the tiny gaps between neurons to transmit thoughts and sensations. Growth factors, which orchestrate the development and repair of tissues, also often operate through this localized, intimate form of communication. For messages that need to travel across vast distances within a multicellular organism, we turn to endocrine signaling. This is the grand global communication network where specialized cells release signaling molecules known as hormones into the bloodstream. These hormones then journey throughout the body, reaching distant target cells that possess the appropriate receptors. Insulin, for instance, released from the pancreas, travels to muscle and fat cells across the body, instructing them to take up glucose from the blood. This is the body's postal service, delivering vital instructions to far-flung destinations. And finally, sometimes cells simply reach out and touch. Direct contact signaling occurs when cells communicate through physical contact, often via molecules embedded in their plasma membranes. Imagine two people holding hands, a direct, undeniable connection. In plants, this happens through plasmodesmatous, tiny channels that directly connect the cytoplasm of adjacent cells, allowing small molecules to pass freely. In animal cells, gap junctions serve a similar purpose, creating direct cytoplasmic bridges for rapid, coordinated responses, particularly in tissues like heart muscle, where synchronized contractions are vital. Once a ligand has bound to its specific receptor, the journey of the signal has only just begun. The information, once external, must now be translated into an internal language the cell can understand and act upon. This process is called signal transduction, a marvel of molecular engineering where the initial signal is converted, amplified, and relayed through a series of intracellular molecules. It's like a whisper becoming a shout, a tiny spark igniting a brilliant cascade. Many receptors, particularly those on the cell surface, are G-protein coupled receptors, GPCRS, or receptor tyrosine kinases, RTKS. When a ligand binds to a GPCR, it causes a conformational change in the receptor, which in turn activates an associated G-protein. This activated G-protein then moves along the membrane to activate another enzyme, often leading to the production of second messengers. These second messengers are small, non-protein, water-soluble molecules or ions that can quickly diffuse throughout the cytoplasm, spreading the signal far and wide. Think of them as nimble internal couriers, rapidly delivering the message to various parts of the cell. Two of the most common and vital second messengers are cyclic AMP, CAMP, and calcium ions, Ca2+. CAMP, for example, is often produced when an enzyme called adenylyl cyclase is activated by a G protein. It then goes on to activate other proteins, particularly protein kinases. Calcium ions, meticulously maintained at very low concentrations in the cytoplasm, can be released from intracellular stores, like the endoplasmic reticulum, in response to a signal, leading to a rapid surge that triggers muscle contraction, neurotransmitter release, or secretion. Another common strategy in signal transduction is the phosphorylation cascade, often orchestrated by protein kinases. Imagine a line of dominoes, each one tipping the next. In a phosphorylation cascade, an initial activated protein kinase uses ATP to add a phosphate group to a specific target protein, thereby activating it. This newly activated protein kinase then phosphorylates another protein, and so on, creating a chain reaction. This process is incredibly powerful because it allows for both amplification. A single activated receptor can lead to the activation of many protein kinases, each phosphorylating many target proteins, and fine-tuning. Each step in the cascade provides an opportunity for regulation, for the signal to be strengthened, weakened, or even diverted, ensuring a precise and context-appropriate response. Ultimately, this intricate dance of reception and transduction culminates in a cellular response. This response can be incredibly diverse, tailored precisely to the original signal and the specific needs of the cell. It might involve changes in gene expression, where specific genes are turned on or off, leading to the synthesis of new proteins or the cessation of old ones. It could trigger the activation or deactivation of specific enzymes, altering metabolic pathways. It might lead to changes in cell shape or movement, as seen in immune cells chasing down pathogens. Or perhaps it prompts the secretion of molecules like hormones or digestive enzymes from the cell. Each response is a carefully orchestrated action, a purposeful fulfillment of the message received. Consider the profound elegance embedded in this system. The just specificity of cellular signaling is breathtaking. Only cells with the correct receptor can hear a particular message, ensuring that insulin acts on muscle cells but not brain cells, or that a growth factor directs its specific target. And the amplification inherent in transduction pathways is nothing short of miraculous. A single molecule binding to a single receptor can unleash a torrent of internal messengers, leading to a massive coordinated cellular response. It is like a tiny ripple expanding into a powerful wave, a quiet thought blossoming into a grand action. As we contemplate the cell as a dynamic communicator, constantly interpreting and responding to the world around it, we begin to appreciate its profound interconnectedness, not just within its own structure, but with the entire tapestry of life. Each cell, a vigilant sentinel, and an eloquent speaker, contributes its voice to the grand biological symphony. This quiet conversation, unfolding moment by moment within and between billions upon billions of cells, is the very heartbeat of existence, a testament to the quiet wonder and profound biochemical elegance that sustains us all. It reminds us that even in the smallest, most hidden corners of life, there is an intelligence, a harmony, and a beauty that transcends our most imaginative dreams. In our last chapter, we marveled at the exquisite ballet of cellular communication, at how molecular keys found their specific locks, igniting cascades of information that ripple through the cell's inner world. We witnessed the whisper of a ligand transformed into a symphony of action, enabling cells to respond, adapt, and coordinate with astonishing precision. But now, as we hold this understanding of individual cellular intelligence, a new profound question gently rises. If each cell is a master communicator, how do these billions, even trillions of individual voices coalesce into the grand living narrative that is you? How do they orchestrate themselves into the magnificent, self-sustaining architectures we call tissues, organs, and entire organisms? We stand at a threshold peering into the very art of biological organization. Imagine, if you will, a vast, bustling city where every single inhabitant possesses the ability to speak, listen, and act. Yet, for this city to thrive, to build its towering structures and maintain its intricate systems, these individual actions must be harmonized, their purposes aligned. This, in essence, is the journey we embark upon now. From the eloquent single cell to the complex cooperative marvel of multicellular life. Let's begin by considering the very first step in this grand assembly. Individual cells, each with its specialized role honed over eons, do not simply exist in isolation within a complex organism. No, they gather, they bond, they form communities. These communities, bound by shared purpose and distinct structures, are what we call tissues. Think of them as specialized guilds within our living city, each dedicated to a particular craft. We observe, for instance, the epithelial tissues, forming protective linings and coverings, like the skin that cradles your form, or the delicate membranes that line your internal organs. Here, cells are often tightly packed, forming impenetrable barriers or selective gates, their surfaces sometimes adorned with microvilli or cilia, each a tiny structure with a specific function. Then there are the connective tissues, the unsung heroes, providing support, binding structures together and filling spaces. Consider the bone that gives you structure, the blood that courses through your veins carrying life, or the adipose tissue that stores energy and insulates. These tissues are characterized by cells often dispersed within an abundant extracellular matrix, a rich tapestry of fibers and ground substance they themselves secrete. Next, we encounter the muscle tissues, the architects of movement. Whether it is the rhythmic beat of your heart, the voluntary contraction of your biceps, or the subtle movements of your digestive tract, it is muscle cells, elongated and rich in contractile proteins, that perform this tireless work. And finally, the nervous tissues, the intricate wiring system composed of neurons that transmit electrical and chemical signals with breathtaking speed, forming the very foundation of thought, sensation, and coordination. Each tissue, a symphony of specialized cells working in concert, is a marvel in itself. But the story deepens, grows richer, for these tissues in turn do not exist independently. They intertwine, they interlace, they collaborate, forming structures of even greater complexity. The organs. Consider your heart, a tireless pump woven from muscle, nervous, and connective tissues, all working in perfect synchronicity to propel life-sustaining blood throughout your body. Or your stomach, a muscular sac lined with epithelial cells, its walls housing nervous tissues that orchestrate digestion, its entire form a testament to the elegant integration of different cellular communities. And the narrative continues its ascent. Organs, each performing a vital function, do not operate in isolation. They unite, they form alliances, creating organ systems. The digestive system, for example, is a complex network of organs: mouth, esophagus, stomach, intestines, liver, pancreas, each contributing its unique function to the grand process of nutrient extraction. The circulatory system, with the heart, blood vessels and blood, forms a vast transportation network. The nervous system, with the brain, spinal cord, and nerves, is the ultimate command and communication center. Here, in this hierarchical organization, from the singular communicating cell to the majestic interconnected organ system, we truly begin to grasp the profound elegance of life's architecture. It is a testament to cooperation, to specialization, and to the emergent properties that arise when individual components unite for a greater purpose. But what truly binds this magnificent structure together, ensuring its continued existence amidst the constant flux of the world? What allows this intricate symphony to maintain its perfect pitch, even when external conditions threaten to pull it out of tune? This brings us to one of the most fundamental and awe-inspiring concepts in all of biology: homeostasis. The word itself whispers of stability, of standing still, yet, in the biological realm, it describes an active, dynamic equilibrium, a continuous, tireless dance of adjustment and recalibration. Homeostasis is the quiet guardian, the internal compass that keeps the myriad biochemical reactions within the body operating within their optimal, often incredibly narrow ranges. It is the unwavering commitment of life to maintain its internal balance, regardless of the turbulent world outside. Consider the delicate balance required for our biochemical machinery. Enzymes, those magnificent protein catalysts we explored, demand specific temperatures and pH levels to perform their duties efficiently. Cell membranes, with their intricate dance of transport and signaling, are sensitive to ion concentrations. Without the vigilant maintenance of these internal conditions, the very fabric of life would begin to unravel. Proteins would denature, metabolic pathways would falter, and the symphony of cellular activity would descend into chaos. So, how does the body achieve this remarkable feat of self-regulation? How does it sense deviations and bring itself back to balance? The answer lies in the elegant design of feedback loops, the very mechanisms that underpin homeostasis. These loops are like the sophisticated control systems of our living city, constantly monitoring, comparing, and adjusting. The most prevalent and vital of these is the negative feedback loop. This is the body's primary way of maintaining stability, of returning a variable to its set point. Imagine a thermostat in a room. When the temperature rises above the set point, the thermostat senses this change and activates the air conditioner to cool the room. Once the temperature drops back to the set point, the air conditioner turns off. This is a self-correcting mechanism. In our bodies, negative feedback loops operate with breathtaking sophistication. Take, for example, the regulation of body temperature. Sensors, specialized nerve cells called thermoreceptors, constantly monitor the internal and external temperature. This information is relayed to a control center, primarily located in the hypothalamus of your brain. If your body temperature rises above the ideal 37 degrees Celsius, 98.6 degrees Fahrenheit, the hypothalamus orchestrates a series of responses. Blood vessels near the skin surface dilate, allowing more heat to escape. Sweat glands are activated, and the evaporation of sweat cools the skin. These are the effectors, carrying out the commands. As the body temperature returns to the set point, these responses are diminished, completing the loop. Conversely, if your temperature drops, the hypothalamus initiates shivering to generate heat and constricts blood vessels to conserve it. Another beautiful example lies in the regulation of blood glucose levels. After a meal, as glucose enters the bloodstream, specialized cells in the pancreas, the sensors, detect this rise. The pancreas then releases insulin, a ligand, a molecular messenger, which acts on various cells throughout the body, signaling them to absorb glucose from the blood and convert it into glycogen for storage. As blood glucose levels fall back to their optimal range, the release of insulin diminishes. Should blood glucose levels drop too low, a different set of pancreatic cells release glucagon, which signals the liver to break down stored glycogen and release glucose back into the blood. This intricate dance, mediated by hormones and their receptors, is a testament to the biochemical precision of negative feedback. Then there is the intriguing, though far less common, positive feedback loop. Unlike its negative counterpart, which seeks to reverse a change, a positive feedback loop amplifies the initial stimulus, driving the system further in the same direction until a specific endpoint is reached. It's like a snowball rolling downhill, gathering more snow and momentum as it goes. A striking example of positive feedback in action is childbirth. As the baby's head presses against the cervix during labor, sensory nerves send signals to the brain. The brain, in turn, stimulates the pituitary gland to release oxytocin. Oxytocin then travels through the bloodstream to the uterus, causing stronger contractions. These stronger contractions push the baby further down, increasing the pressure on the cervix, which then sends more signals to the brain, leading to the release of more oxytocin and thus even stronger contractions. This amplifying loop continues, intensifying the process until the baby is born, at which point the stimulus is removed and the loop ceases. Another familiar instance is blood clotting. When a blood vessel is damaged, platelets adhere to the injury site. These activated platelets release chemicals that attract more platelets, which then release more chemicals, creating a rapidly growing plug that seals the wound. This cascade of amplification ensures a swift and effective response to injury. It is truly humbling to consider how these biochemical principles, from the specificity of a ligand binding to its receptor, to the catalytic power of an enzyme, to the energy currency of ATP, are all woven into the very fabric of these feedback loops. Hormones act as ligands, signaling target cells. Enzymes catalyze the reactions that produce or break down these hormones, or the substances they regulate. ATP powers the pumps and processes involved in cellular responses. The entire orchestra of biochemistry, which we have been so carefully exploring, is precisely what allows these intricate homeostatic mechanisms to function, maintaining the quiet persistence of life. So, as we reflect upon the journey from the atom's silent dance to the cell's vibrant communication, and now to the organism's elegant self-regulation, we are invited to a deeper appreciation. We see that life is not merely a collection of parts, but a dynamic, interconnected system, constantly striving for balance, constantly adapting. Homeostasis is not a static state, but a dynamic flow, a continuous conversation between the internal and external worlds, ensuring that the symphony of life plays on, harmonious and enduring. It is a quiet testament to the resilience and profound intelligence embedded within every living being, a constant whisper of wonder in the grand tapestry of existence. The quiet persistence of life we discovered in our last journey is a testament to an exquisite, tireless self-regulation, a concept we gently named homeostasis. It is the body's unwavering commitment to balance, a constant subtle recalibration amidst the ceaseless flux of existence. We spoke of negative feedback loops, those gentle hands that guide us back to equilibrium, and the rarer, amplifying whispers of positive feedback. But how, we might quietly ask, does this magnificent, intricate dance truly unfold? What unseen choreography keeps the delicate systems of our inner cosmos in such harmonious motion, moment by precious moment? Today, we delve deeper into this profound question, exploring the very heart of how life maintains its equilibrium, not as a static state, but as a dynamic, living process. Imagine, if you will, a vast complex orchestra playing a continuous, intricate symphony. Each instrument, each musician, is constantly adjusting, listening, responding, ensuring that the overall melody remains pure, vibrant, and alive. This is the essence of homeostasis at its molecular core, a ceaseless, energy-intensive endeavour powered by the very same biochemical principles we have so lovingly explored. Consider first the sheer energy cost of this tireless vigilance. To maintain temperature, to regulate pH, to precisely manage the concentrations of countless ions and molecules, to repair, to rebuild, to respond. All these acts of self-preservation demand a continuous, substantial supply of energy. It is not a passive drifting towards balance, but an active, deliberate push against the entropy that would otherwise pull us apart. Our cells, those bustling metropolises we explored, are ceaselessly burning fuel, generating the universal currency of ATP, not merely for grand tasks like movement or thought, but for the quiet, persistent work of maintenance. Think of the mitochondria, those ancient powerhouses, as the heart of this energetic commitment. Within their folded membranes, the exquisite dance of cellular respiration unfolds, tirelessly converting the simple sugars and fats we consume into the packets of energy that fuel every single regulatory mechanism, every enzyme that adjusts a metabolic pathway, every protein channel that opens or closes to manage an ion gradient, every receptor that senses a subtle shift in the internal environment. Each requires ATP to perform its essential role. It is a constant, quiet hum of energy production, an unseen river of vitality flowing through every cell, ensuring that the symphony of life never falters. This, dear friends, is the bedrock upon which the entire edifice of homeostasis rests, an unyielding, energetic dedication to self-preservation. But how precisely does the body know when a change has occurred, when the internal currents begin to drift from their optimal flow? Here we encounter the magnificent network of molecular sentinels and messengers, the exquisite sensitivity woven into the very fabric of our being. Think back to our discussions of cellular signaling, of ligands and receptors, of the intricate protein locks and keys that orchestrate communication. These are the very same players, now imbued with the specific vital task of monitoring our internal milieu. Imagine tiny dedicated sensors, often protein receptors embedded in cell membranes or floating within the cytoplasm, each exquisitely tuned to a particular parameter. There are receptors that bind to glucose, signaling its abundance or scarcity. There are ion channels that open or close in response to minute changes in electrical potential or the presence of specific ions. There are enzymes whose activity is finely modulated by shifts in pH or temperature. These molecular sentinels are not merely passive detectors, they are active interpreters, translating a physical or chemical change into an immediate biochemical signal. When a sentinel detects a deviation, perhaps a slight rise in blood acidity, or a dip in oxygen levels, it initiates a cascade, a whisper that echoes through the cellular landscape. This whisper is often a change in the sentinel's own conformation, a subtle twist or turn in its three-dimensional structure, which then triggers the next step in the signaling pathway. Perhaps it activates a G protein, or initiates a phosphorylation cascade, or releases a burst of second messengers like cyclic AMP or calcium ions. These are the molecular messengers, relaying the urgent news, amplifying the signal as it travels deeper into the cell. Consider, for instance, the delicate regulation of blood glucose. When you enjoy a sweet treat, glucose levels in your blood rise. Specialized cells in your pancreas, acting as vigilant sentinels, detect this increase. Their internal machinery, sensitive to the influx of glucose, responds by releasing insulin, a hormonal messenger. Insulin then travels through the bloodstream, a molecular envoy, binding to specific receptors on target cells throughout the body. Muscle cells, fat cells, liver cells. This binding acts as a key, unlocking a cascade of events within these cells, prompting them to absorb glucose from the blood, perhaps converting it into glycogen for storage. As glucose levels return to their optimal range, the pancreatic sentinels detect this return to balance, and the release of insulin diminishes. It is a beautifully orchestrated feedback loop, a quiet conversation between cells, all working to maintain that perfect, life-sustaining equilibrium. This, then, is the symphony of response, a molecular orchestra where each section plays its part in restoring harmony. Once a signal of imbalance is detected and relayed, the cell, or indeed the entire organism, initiates a series of precise adjustments. These responses are incredibly diverse, yet all are aimed at neutralizing the disturbance. For example, if our internal thermometer detects a drop in core body temperature, the molecular symphony begins. Hormonal messengers might stimulate increased metabolic activity, causing cells to generate more heat. Muscles might begin to shiver, an involuntary contraction that generates warmth. Blood vessels near the skin surface might constrict, minimizing heat loss to the external environment. Each of these macroscopic responses is underpinned by a myriad of microscopic biochemical events. Changes in enzyme kinetics, alterations in gene expression, leading to the synthesis of new proteins, adjustments in membrane permeability, or shifts in the activity of specific metabolic pathways. Think of the exquisite control over blood pH, a parameter so vital that even slight deviations can be catastrophic. Our blood, with its delicate buffering systems, acts like a chemical sponge, absorbing excess acids or bases. But beyond this immediate chemical defense, our kidneys and lungs play crucial roles, finely tuning the excretion of acids or the retention of bicarbonate ions. At the cellular level, this involves complex transport proteins, enzymes that catalyze the formation or breakdown of bicarbonate, and intricate signaling pathways that inform these organs of the precise pH status. It is a continuous moment-by-moment adjustment, a testament to the profound interconnectedness of our biochemical systems. This intricate dynamic process also reveals a deeper truth about homeostasis. It is not always about rigidly maintaining a single fixed set point. Life, in its profound wisdom, also possesses the capacity for allostasis, maintaining stability through change. Rather than simply returning to a predetermined norm, the body can adapt its set points in response to sustained challenges or shifting environmental demands. Imagine a long-distance runner training for a marathon. Over weeks and months, their body adapts. Their cardiovascular system becomes more efficient. Their muscles develop greater endurance. Their metabolic pathways become more adept at utilizing different fuels. These are not merely temporary adjustments. They represent a fundamental adaptive recalibration of their homeostatic set points, allowing them to function optimally under increased physiological stress. Their quote 20 might shift, expanding the range within which their systems operate comfortably. This is allostasis in action, a flexible resilience, a proactive adjustment of internal parameters to anticipate and meet future demands. It is the body's way of saying, I will not just react, I will prepare. This adaptive capacity highlights the profound intelligence embedded within our biochemistry, the ability to learn, to adjust, to evolve its internal architecture to better navigate the currents of existence. It is a testament to the dynamic, living nature of our internal balance, far from a simple thermostat, but rather a sophisticated self-optimizing system. Yet despite this magnificent choreography, this tireless vigilance, there are moments when the rhythm falters. Life, in its inherent fragility, can experience imbalance. When the delicate feedback loops are overwhelmed, when the molecular sentinels are damaged, or when the energetic demands outstrip supply, homeostasis can be compromised. This is where we begin to glimpse the origins of disease, not as an external attack, but often as an internal disruption to this exquisite balance. Consider the prolonged effects of stress, for example. While acute stress responses are vital for survival, chronic stress can lead to a sustained elevation of stress hormones, pushing the body's systems beyond their adaptive capacity. The allostatic load, the corticosteroid on the body from repeated or chronic stress, can eventually lead to a breakdown in regulatory mechanisms, contributing to conditions like hypertension, metabolic disorders, or compromised immune function. The molecular symphony, once harmonious, begins to play out of tune, leading to a state of dis-ease. Or think of diabetes, where the exquisite control over blood glucose falters, either due to insufficient insulin production or the body's inability to respond effectively to insulin signals. The molecular sentinels are either silent or unheard, and the vital feedback loop is broken, leading to a cascade of cellular and systemic consequences. These moments of imbalance serve as poignant reminders of the fragility and preciousness of the homeostatic dance, underscoring the vital importance of every molecular interaction, every enzyme, every receptor in maintaining the quiet wonder of being alive. As we conclude this exploration, let us pause once more to appreciate the unseen choreography unfolding within us every second of every day. From the tireless engines of our mitochondria, generating the energy for life, to the vigilant sentinels detecting the faintest shifts in our internal landscape, to the molecular orchestra playing its symphony of response, and the profound capacity for adaptive resilience. All of it is a testament to the breathtaking elegance of biochemistry. It is a constant, quiet miracle, a continuous act of self-creation and self-preservation, ensuring that the flame of life continues to burn brightly, balanced on the delicate edge of existence. In this profound balance, this ceaseless, intricate dance, we find not just the mechanics of life, but a deeper reverence for the very fabric of being. We have journeyed together through the intricate architecture of life, from the quiet dance of atoms forming bonds to the grand symphony of macromolecules, the bustling metropolis of the cell, and the exquisite ballet of cellular communication. We have witnessed the tireless efforts of homeostasis, the vigilant guardian that seeks to maintain a delicate balance, a serene equilibrium amidst the constant flux of existence. We understand that when this intricate system falters, when the feedback loops become overwhelmed, the harmony can break and disease may emerge. But consider for a moment the deeper mystery that underpins even this remarkable self-regulation. How does this intricate blueprint, this whisper of instructions, persist across generations? How does the quiet wonder of being alive find its unique expression in each individual, and yet retain the fundamental essence of its lineage? We arrive now at a profound truth, an elegant continuation of our exploration, the very heart of inheritance, the echoes of life's instructions that resonate from the deepest past into the unfolding future. We return to the molecule we first encountered as a nucleic acid, a polymer of nucleotides, deoxyribonucleic acid, or DNA. We spoke of it then as a vast molecular library, a repository of information. Now, let us truly open its pages, for within its elegant double helix lies the master blueprint, the comprehensive instruction manual that guides the construction, operation, and even the subtle variations of every living being. Imagine DNA not merely as a chemical compound, but as an ancient sacred text, inscribed in a language of 4 letters, A, T, C, G. These letters, arranged in countless permutations along the 2 spiraling strands, form words, sentences, and paragraphs, each a gene, a specific instruction for a particular task. This sacred text is housed within the nucleus, a molecular vault, protected and cherished. It is a testament to life's foresight, its ingenious solution for storing an unimaginable wealth of information with remarkable stability. The double helix, with its complementary pairing, offers an inherent fidelity, a built-in mechanism for self-correction, an accurate replication, ensuring that these vital instructions are passed down with astonishing precision. But a blueprint, however perfect, is inert without a builder. A sacred text, however profound, remains silent without a reader and a translator. And so, we delve into the marvelous process by which these silent instructions are brought to life, a journey often referred to as the central dogma of molecular biology. It is a 3-act play, a graceful flow of information from DNA to RNA, and finally to the proteins that perform the vast majority of life's work. The first act is transcription, a quiet, meticulous process akin to a diligent scribe making a working copy from a priceless manuscript. Within the nucleus, specific segments of the DNA blueprint, individual genes are identified. Here, an enzyme, RNA polymerase, acts as our scribe. It unwinds a small section of the double helix, gently separating the 2 strands. Then, using one of these DNA strands as a template, it synthesizes a complementary strand of messenger RNA, or mRNA. This mRNA molecule is not the original, but a faithful single-stranded transcript ready to carry its message out into the bustling cytoplasm. It is a temporary, mobile instruction sheet, carefully crafted for immediate use, leaving the precious DNA master copy undisturbed and safe within its nuclear sanctuary. Once transcribed, this mRNA molecule embarks on its journey, exiting the nucleus through tiny pores and venturing into the wider cellular landscape. It seeks out the bustling factories of the cell, the ribosomes, where the second, even more intricate act of our play unfolds: translation. Imagine the ribosome as a sophisticated molecular assembly line, a tireless artisan. The mRNA molecule arrives, its sequence of nucleotide, 23, carrying the coded message. But how does the cell translate this 4-letter language into the 20-letter language of amino acids, the building blocks of proteins? This is where the exquisite elegance of the Sirstian genetic code comes into play. Each sequence of 3 mRNA nucleotides, known as a codon, specifies a particular amino acid. It is a universal language, spoken by nearly all life forms on Earth, a testament to our shared biochemical ancestry. As the mRNA threads its way through the ribosome, another crucial player enters the scene: transfer RNA, or tRNA. These small, cloverleaf-shaped molecules are the molecular interpreters. Each tRNA carries a specific amino acid at one end, and at the other, a 3-nucleotide sequence called an anticodon, which is complementary to an mRNA codon. It is a perfect lock-and-key mechanism, ensuring that the correct amino acid is delivered precisely when needed. The ribosome orchestrates this incredible ballet. It reads the mRNA codons one by one. As each codon is read, the corresponding tRNA molecule bearing its specific amino acid docks temporarily with the mRNA. The ribosome then catalyzes the formation of a peptide bond between the incoming amino acid and the growing chain of amino acids, slowly, steadily, building the protein, link by precious link. It is a marvel of precision, a rapid-fire assembly where hundreds, even thousands of amino acids are joined in a specific order, creating a polypeptide chain that will soon become a functional protein. And what happens to this newly synthesized polypeptide chain? We return to a familiar theme: folding. We spoke of proteins finding their unique 3-dimensional shapes, a process crucial for their function. The nascent polypeptide emerging from the ribosome is not yet a working protein. It must fold, often with the assistance of molecular chaperones, into its intricate secondary, tertiary, and sometimes quaternary structures. It is only then, when it achieves its precisely sculpted form, that it can truly become an enzyme, a structural component, a transporter, a signaling molecule, or any of the myriad roles we explored before. Thus, the journey from DNA blueprint to active protein is complete, a testament to the profound interconnectedness of all biochemical processes. But the story of life's instructions is not simply about reading the blueprint. It is also about knowing when and where to read it. Not every gene is active in every cell, nor at every moment. Consider the profound specialization within your own body. A nerve cell expresses a different set of genes than a muscle cell or a skin cell, even though every cell contains the same complete DNA blueprint. This remarkable selectivity is thanks to the intricate process of gene expression regulation. Imagine the blueprint having a complex system of molecular switches and dimmer controls. These regulatory mechanisms determine which genes are "24" or "25" and to what extent they are expressed. Proteins called transcription factors act as molecular sentinels, binding to specific DNA sequences near genes, either promoting or inhibiting their transcription. There are also more subtle yet powerful layers of control, often referred to as epigenetics. These are not changes to the DNA sequence itself, but rather chemical "26" on the DNA or its associated proteins, histones, that can influence how tightly the DNA is packed, and thus how accessible it is to the transcriptional machinery. It is as if certain pages of the blueprint are gently highlighted or subtly obscured, making them easier or harder to read without altering the original text. This dynamic regulation allows cells to adapt to changing environments, to specialize, and to coordinate their functions within the vast organism. And yet, even with all this precision and regulation, life is not static. No blueprint is copied perfectly forever, and this is where the profound beauty of variation enters our story. Occasionally during DNA replication or due to environmental factors, a "27" occurs, a change in the DNA sequence. We call these changes mutations. For a long time, mutations were often viewed solely as errors, as sources of disease or defect. But in the grand tapestry of life, mutations are also the very engines of change, the wellspring of diversity. These subtle alterations in the genetic code give rise to different versions of genes, which we call alleles. It is these alleles that account for the unique characteristics that make each individual distinct, from the color of your eyes to your predisposition to certain traits, or even your susceptibility to certain diseases. Most mutations are neutral, having no significant effect. Some can be harmful, leading to the breakdown of essential protein function, and, potentially, to disease. But crucially, some mutations can be beneficial, providing an organism with a new advantage, a novel adaptation that allows it to thrive in a changing world. It is through the accumulation and selection of these beneficial variations over vast stretches of time that life has been able to diversify into the astonishing array of species we see today. This brings us to the grand culmination of our understanding, the link between genotype and phenotype. Your genotype is your unique genetic makeup, the sum of all the alleles you carry, your personal inherited blueprint. Your phenotype, on the other hand, is the observable expression of that blueprint, your physical traits, your biochemical characteristics, even many aspects of your behavior. It is the visible manifestation of the intricate molecular dance within. The journey from genotype to phenotype is not always a simple, one-to-one correspondence. It is a complex interplay, a continuous dialogue between your inherited instructions and the environment in which you develop and live. Nutrition, exposure to toxins, social interactions, even the subtle cues from your surroundings. All these can influence how your genes are expressed, shaping the final phenotype. It is a constant dynamic interaction, a beautiful testament to the fluidity of life. Your biochemistry is not a fixed entity, but a living, breathing symphony, constantly adapting and responding. As we reflect upon this journey from the silent blueprint to the vibrant expression of life, we uncover a profound truth. Life is not merely a collection of molecules, but a self-perpetuating, self-correcting, and self-evolving system. It possesses the astonishing capacity to store its own instructions, to meticulously execute them, to regulate their expression with exquisite precision, and crucially, to introduce subtle variations that allow for adaptation and diversification across eons. This elegant system of inheritance ensures that the quiet wonder of biochemistry, the intricate dance of atoms and molecules, continues its timeless performance. It allows for the persistence of life's fundamental principles, while simultaneously providing the creative spark for novelty and evolution. It is a humbling thought, isn't it? That within the double helix, within the very fabric of your being, lies an ancient whisper, an unbroken chain of instructions stretching back to the dawn of life, continuously adapting, continuously unfolding, a testament to the enduring, awe-inspiring power of biochemistry. And with this profound understanding, we are ready to consider how these living systems gather and utilize the very energy that fuels this endless, wondrous cycle. We have journeyed through the intricate blueprints of life, tracing the elegant helix of DNA, understanding its silent commands, and witnessing its transformation into the myriad forms of proteins that give rise to all observable traits. We've seen the molecular architects at work, the cellular cities bustling with communication, and the delicate dance of homeostasis maintaining a vibrant equilibrium. Yet, a blueprint, however perfect, remains but a potentiality without the force to bring it to life, without the ceaseless hum of activity that powers every whisper, every thought, every beat of existence. So as we stand at this threshold, a profound question beckons. Where does the energy come from? What fuels this endless, wondrous cycle of life? This constant renewal? This tireless striving against entropy? It is a question that leads us into the heart of metabolism, into the very engines that drive the living world, from the smallest bacterium to the grandest redwood, to you and me. Imagine, if you will, the universe as a vast ancient clock, wound by an unseen hand. Life, in its magnificent ingenuity, has learned to harness the unwinding of that clock, to capture its subtle movements and transform them into the vibrant pulse of being. At its most fundamental, our energy begins with the most primal and generous star in our sky, the sun. Consider the sun, a distant fiery heart pouring forth its radiant energy across the vast emptiness of space. This light, this pure energy, is the ultimate wellspring for nearly all life on Earth. And here we encounter one of life's most poetic acts of alchemy, photosynthesis. It is the silent, verdant breath of our planet, performed by plants, algae, and certain bacteria. They are the primary architects of energy, the quiet dreamers who gather the sunbeams and weave them into the very fabric of organic molecules. In the chloroplasts, those verdant jewel-like organelles within plant cells, a symphony unfolds. Water is drawn from the earth, carbon dioxide from the air, and with the gentle kiss of sunlight, the simple ingredients are transformed. Light energy is captured, much like a butterfly caught in a net, and then carefully, meticulously, it is stored within the chemical bonds of glucose, a simple sugar. Oxygen, a precious byproduct, is released, becoming the very breath we inhale. This glucose, this stored sunlight, is more than just a sugar. It is a compact parcel of potential, a miniature battery charged by the cosmos. It is the first grand currency of energy, passed from the sun-kissed leaf to the hungry herbivore, and then up the intricate ladders of food webs, eventually reaching us. We, like all consumers, cannot directly capture the sun's brilliance. Instead, we are inheritors of its legacy, partaking in the energy gifts bequeathed by the photosynthetic world. And so, we arrive at the intricate process of cellular respiration, the grand metabolic pathway through which living cells extract the energy stored in organic molecules, primarily glucose, and convert it into ATP, our universal energy currency. If photosynthesis is the slow, deliberate act of charging a battery, cellular respiration is the controlled, elegant discharge, powering every blink, every thought, every beat of the heart. It is a multi-stage journey, an odyssey within the cell, primarily unfolding in the cytoplasm and the mitochondria, those bustling powerhouses we have explored before. Let us embark on this journey and witness the meticulous extraction of energy. Our story begins in the cytoplasm with a 6-carbon molecule of glucose. Here, an ancient pathway, conserved across eons of evolution, takes center stage. Glycolysis. The word itself whispers its purpose. Quote, 28. Imagine glucose as a precious gem and glycolysis as the initial careful cleaving. Through a series of 10 enzymatic steps, this single glucose molecule is gently broken down into 2 molecules of pyruvate, each containing 3 carbons. This initial phase is a beautiful paradox. It requires a small investment of ATP to begin, like priming a pump, but it yields a modest net gain of 2 ATP molecules and 2 molecules of NADH. NADH, you recall, is an electron carrier, a molecular taxi laden with high-energy electrons, ready to transport its precious cargo to the final, most productive stage of energy generation. Glycolysis is swift, an anaerobic dance that can occur even in the absence of oxygen, a testament to its primordial origins, providing a quick burst of energy when needed. From the bustling cytoplasm, our 2 pyruvate molecules, now lighter and more dynamic, journey into the inner sanctum of the mitochondria, crossing its protective membranes. Here the journey takes a deeper turn, preparing for the grand extraction. Each pyruvate molecule undergoes a transformation known as pyruvate oxidation. It's like a final refinement before entering the main forge. A carbon atom is removed, released as carbon dioxide, a molecule we exhale, a subtle echo of the fuel we consume. The remaining 2 carbon molecule, now called an acetyl group, is attached to a coenzyme A, forming acetyl-CoA. More NADH is produced here, adding to our growing fleet of electron carriers. Now, with acetyl-CoA in hand, we enter the heart of the mitochondrial matrix into a cyclical marvel known as the Krebs cycle, or the citric acid cycle. Envision a whirling carousel, a metabolic hub where the acetyl group is systematically dismantled, its remaining energy-rich bonds carefully broken. This cycle is a masterpiece of biochemical engineering, turning twice for each original glucose molecule, once for each acetyl-CoA. In each turn of the Krebs cycle, the 2-carbon acetyl group combines with a 4-carbon molecule forming citrate, hence the citric acid cycle. Through a series of 8 enzymatic reactions, citrate is then progressively oxidized, releasing 2 more molecules of carbon dioxide, the last of the original glucose carbons, and generating more electron carriers, 3 NADH molecules and 1 FADH2 molecule. A small amount of ATP, or GTP, which is quickly converted to ATP, is also directly produced. The true treasure of the Krebs cycle, however, lies not in the direct ATP it yields, but in the abundant harvest of NADH and FADH2. These are the unsung heroes, the diligent couriers carrying the vast majority of the glucose's captured energy in the form of high-energy electrons. They are poised to deliver their cargo to the final, most spectacular act of cellular respiration. This brings us to the inner mitochondrial membrane, a highly folded landscape that dramatically increases its surface area, allowing for the grand finale. Oxidative phosphorylation. This stage is where the bulk of ATP is generated, a process that breathes life into the very term powerhouse. Imagine a molecular waterfall, a cascade of electrons tumbling down a series of protein complexes embedded within this inner membrane. This is the electron transport chain. NADH and FADH to arrive, releasing their high-energy electrons. As these electrons move from one protein complex to the next, they gradually lose energy, much like water flowing downhill. This liberated energy is not wasted. It is ingeniously used to pump protons, H+ ions, from the mitochondrial matrix into the intermembrane space, creating a steep concentration gradient, an electrochemical potential difference. This proton gradient is the true genius of oxidative phosphorylation. It is a form of stored energy, much like water held behind a dam. The protons, yearning to flow back into the matrix where their concentration is lower, find only one path, through a remarkable molecular turbine called ATP synthase. As protons rush through the ATP synthase channel, they spin a tiny rotor within the enzyme, much like water turning a hydroelectric turbine. This mechanical energy is then harnessed to catalyze the synthesis of ATP from ADP and inorganic phosphate. This process, known as chemosmosis, is an astonishing feat of molecular engineering, generating approximately 26 to 28 ATP molecules per glucose molecule. It is the culmination of the entire journey, the grand conversion of chemical energy into the immediate fuel that powers virtually every cellular activity. In total, from one single molecule of glucose, the entire process of cellular respiration yields a magnificent bounty of approximately 30 to 32 ATP molecules. This is the energy that allows DNA to replicate, proteins to fold, signals to transmit, muscles to contract, and thoughts to form. But what if glucose isn't available? What if our cells need energy from other sources? This is where the beauty of life's interconnectedness truly shines. The metabolic pathways are not rigid, isolated roads. They are a vast interwoven tapestry, a network of highways and byways. Consider the lipids, those long-term energy stores we discussed. When carbohydrates are scarce, our cells can tap into fat reserves. Triglycerides are broken down into glycerol and fatty acids. Glycerol can be converted into an intermediate of glycolysis, while fatty acids are cleaved into 2 carbon units that become acetyl-CoA, directly feeding into the Krebs cycle. This is why fats are such efficient energy stores. Their long hydrocarbon chains yield a tremendous amount of acetyl-CoA, and thus a vast quantity of ATP, even proteins. Those versatile molecular machines can be pressed into service as an energy source, though this is typically reserved for times of starvation or when protein intake is excessive. Amino acids, after their amino groups are removed, a process that produces ammonia, which must then be safely excreted, can be converted into intermediates of glycolysis or the Krebs cycle, joining the energy-generating flow. This intricate web of pathways highlights the adaptability and resilience of living systems. The cell is a master alchemist, capable of transforming one form of fuel into another, always striving to maintain its energy supply, always seeking equilibrium. And who orchestrates this complex ballet of energy transformation? It is the cell itself, through a sophisticated network of regulatory mechanisms. Enzymes, those tireless catalysts, are the key points of control. Feedback inhibition, allosteric regulation, and hormonal signals constantly fine-tune the rates of glycolysis, the Krebs cycle, and oxidative phosphorylation. When ATP levels are high, the pathways slow down, conserving resources. When energy is needed, the pathways accelerate, ensuring a steady supply. It is a testament to the cell's quiet wisdom, its ability to sense its own needs and respond with elegant precision. As we conclude this exploration of energy, let us pause and reflect on the profound implications. Every breath we take, every beat of our heart, every thought that flickers through our minds is powered by this ancient, elegant dance of molecules, a dance that began with the distant light of our sun. From the silent, sun-kissed leaf to the bustling inner membrane of a mitochondrion, energy flows, transforms, and sustains. It is a humbling realization, connecting us to the very fabric of existence, to the deep time of the universe, and to every other living creature that shares this planetary journey. We are not merely observers, we are participants in this grand cosmic energy cycle. The quiet wonder of being alive, the very essence of our vitality, is woven into these biochemical pathways, a continuous testament to the ingenuity and interconnectedness of life. May this understanding deepen your quiet curiosity, inviting you to see the world not just as it appears, but as the magnificent, energy-driven symphony it truly is.